Hello, everyone, and welcome to today's seminar, Revealing Biological Complexity Through Spatial Profiling. Thank you all for joining us. My name is Ramalakshmi Ramasamy, and I'm a postdoctoral associate in Dr. Paul Robson's lab at the Jackson Laboratory for Genomic Medicine. It's my pleasure to introduce Dr. Fei Chen. Dr. Chen is an assistant professor in the Department of Stem Cell and Regenerative Biology at Harvard and a core faculty member at the Broad Institute. His lab develops innovative tools at the intersection of microscopy, genomics, and synthetic biology to bridge single-cell genomics with spatial and temporal information. By enabling researchers to map where cell types are located within intact tissues and when key transcriptional programs are active, his work is helping uncover fundamental principles of tissue organization in development as well as mechanisms of disorganization in injury and disease. Today, he will be speaking about spatiotemporal genomics linking cell history to tissue architecture. Dr. Chen, the floor is yours.
Okay, yeah, so thanks for the introduction. And I thought today I would give an overview of some of the technologies that we're using in the group, but also how we're thinking about problems of spatial biology through highlighting three very short examples. And so this is an H&E picture of a cell. It's actually hard to make out the details. But actually, when you zoom out, you can actually see this. This is the cell in the context of tissue. It's actually a Hodgkin lymphoma tumor cell. You can see there's this large nucleus surrounded by cells of varying types and shapes and sizes. And you know, these large cells are kind of like sparsely distributed throughout the tissue. And these details might be important for like maybe a pathologist trying to make a diagnosis. I highlight these details not to point out the difficulty interpreting zoomed in microscopy images, but to make an analogy. I mean, oftentimes what we're doing in genomics is studying dissociated cells, kind of zooming in on that one cell that we had in there. And we become really good at studying single cell properties like their transcriptomes, their genomes, and their epigenomes. And so probably life would be good if we were all just single cells. But in humans and multicellular organisms, the spatial context is quite important for understanding tissue and to understand biology. And space is then a frontier of single cell genomics. And my lab has been working on this challenge. And we've been doing this for a while. So over the past few years, actually funded by HETD and HERI, we've built many tools to measure the transcriptomes, genomes, and other features directly in tissues, building on top of array-based approaches that capture these molecules in spatial context. Building on these, we've recently developed what we believe to be kind of a uniquely enabling technology for spatial genomics called slide tags. And it's a way to a universal way to perform single-cell sequencing with spatial context that we published a couple of years back. And I'll just quickly explain how that works. The concept between slide tags is pretty straightforward. You can perform your single-cell sequencing and know where your cells came from in the tissue. To achieve this, what happens is that tissues are placed on an array of barcoded beads, each with a unique DNA sequence marking its XY location. These barcodes diffuse into the cells, actually, and attach into their nuclei. You take section frozen tissue onto the array, and then we can photocleave these barcodes, and then they can diffuse into the nuclei. And then when sequencing is performed, what's actually happening is that each cell is picking up the barcodes from nearby beads, and if this reveals the spatial origin of each cell, essentially acting as like a GPS for cells. And this enables, when you perform single cell measurements, it allows single cell measurements to have all of the spatial information. And so what does that data actually look like? This is one of the first slide tags experiments we did in the mouse hippocampus. Here is a UMAP representation of different types of cells where each dot represents a single cell and the clusters represent the principal cell types. But if you look at this green cell, here are the spatial barcodes it picked up in spatial coordinates, and you can see that it picked up this little cluster of barcodes at maybe x equals 1,000 and y equals something. And here's the marginal distribution in x and y, and you can fit the localization of the cell with quite high accuracy. And you can do that with almost all of these cells in space, and then you can reconstruct the architecture of the tissue. Here, you can, if you spatially map all the cells, you show site tags recapitulates the expected tissue architecture, including the underlying gene expression of, what each one of these cells samples the transcriptome, and it's very high quality transcriptomic data, thousands of UMIs per cell across all 20,000 genes. So there's many technical details and demonstrations in the manuscript, but I don't want to spend too much time on that since it's already published. I just want to highlight the key takeaways, and then we'll get to the examples of how we actually think about the data. Importantly, it retains the resolution and data quality of single cell sequencing while enabling measurements at about 10 micron resolution. And in fact, the resolution doesn't really matter because it's true single cell data. And it allows for kind of addresses some key limitations that allows for the integration of any single cell technology into spatial context. And recently, as part of our NHGRI effort, we've been doing FFPE RNA, as well as DNA whole genome and then DNA methylation. And I won't talk about that today because I wanted to really highlight how we think about applying these technologies to biological problems, but I'm happy to answer any of these questions. So then thinking about the problems, so how do we actually use this sort of technologies, these sort of spatial technologies to answer biological questions? And our goal is to understand how cells and tissues are organized and how their states are influenced by their environment. And you can think of that in a couple of questions, right? Like what is the organization of a tissue in terms of molecularly defined cell types? What are your neighborhoods and interactions? And then ultimately, how do you communicate to your neighbors via signaling molecules? And then ultimately, how does that shape back to changing your cells, your internal transcriptomic state? And so to highlight how we think about it, I'll just highlight three vignettes, one for each of these: so the organization, the interactions, and communications. So for organization, this is a study that we did a while back, not actually on slide tags, but on the precursor technology slide-seq, which is spatial transcriptomic technology. But the concepts are the same. And here we try to answer how's the mammalian brain organized by molecularly defined cell types. And what we did was we sought out to actually build a complete cell type atlas in the mouse brain. My collaborator, Evan McCosco, and we first took a mouse brain and performed 100 anatomical dissections, generating about 6 million single cell profiles, which we can then cluster into this UMAP you see here. And in parallel, actually, Evan Murray, who has a research science lab, and Carol, an RA in Evan's lab, performed 101 serosections of spatial transcriptomics on one brain. They had to be very careful not to mess up, and then we can reconstruct this organization of the architecture of cell types in the entire brain. And there's a lot of data in the paper, but maybe just a couple of like key highlights and takeaway is that here is actually very interesting. So there's, if you look at these 6 million cells, there's about 5,000 different cell types, the majority of which are neurons. The majority of the types, 50% of the cells are glia, but most of the diversity is in neurons. And here is the anatomical regions of the brain split by types of cells as localized by spatial transcriptomics. And you can see this strong diagonal effect. And that just means that most neurons are incredibly localized. They're very focal. They're mostly only localized to one or two of these brain regions. And you can see that here, any given UMAP cluster, for example, here are a couple of examples mapped to a very specific region of the brain. And another kind of interesting and very surprising thing is that if you look at where the diversity is, these three boxes represent the hippocampus, the cortex, and the cerebellum, three traditionally very well-studied regions of the brain. That's not where a lot of the cell type diversity is. It was overwhelmingly localized in what we would call like the brainstem, in the hypothalamus, brainstem, and midbrain. These are areas of the brain we think of as serving very basic human functions, like regulating alertness, reflexes, movement, and appetite. I think it's very, this the majority of what we think of neuro many people choose to study the cortex and these other parts because these are the parts that has heavily expanded in humans and maybe implicated in intelligence and such. But I think there's a lot of very quite intricate nuggets in the hypothalamus and midbrain, and in fact, Evan and I thought about this, what this meant for a long time. Why these atlases reveal the surprising amount of diversity in the brainstem and hypothalamus, and actually we think there's this very interesting analogy to machine learning, like not like it's like machine learning as we think about like AI systems being developed, in the sense that actually these meant there are parts of the brain that represent kind of the learning subsystem where there are very few cells types, but there's many copies of those types. Like you can think of the cortex as like just repeated copies of this particular computational layer. And if you want to scale how much computation you want to do, you can just keep on repeating that motif, and but you don't necessarily need to diversify the types of cells. But then, if you think about how we train ML models, perhaps, and that's kind of like building a neural network. And if you think about how we train ML models, we often have to give it objective functions and training data and algorithms. And actually, you can think of these other parts of the brain as like the steering subsystem, which is like these objective functions, for example, or boutique rules about how an organism should behave in any given context. And there you can encode that into the genome and then subsequently into the animal through boutique and unique cell types. All right, so that's how the brain is molecularly organized. Next is maybe let's talk about interaction. So if tissue architecture shapes normal biology, what about disease, and how does tissue context change in disease? And so I'll highlight a story in the tumor microenvironment and an immune system, which are great systems for exploring these questions as cell interactions really influence cell behavior. We performed actually slide tags here on melanoma, metastatic melanoma. And here you can actually see the spatial maps of types of cells, spatial single cells. And then here is the UMAP. So these actually directly are aligned. And interestingly, if you just look at, these are the tumor clusters, and for example, here are the immune clusters and stromal clusters. If you just look at the human tumor clusters, you can see that they're each defined by there's three types that are defined by unique transcriptomic states. When mapped spatially, these states are segregated, aligning closely with anatomical, so like histological regions of the tumor. And then actually we wanted to understand, so those are kind of neighborhoods. And so we actually wanted to understand if these transcriptional states were driven by genetic differences. And so using genome-wide data, we can actually analyze copy number variation across single cells, shown here on the rows, with red representing gains and blue representing losses on chromosomes. And you can see that these transcriptome states actually map to shared genomic alterations, suggesting some common ancestral clone, but then also unique changes in these genetic subclones. But I alluded that we wanted to study the interactions of these tumor cells with their environment. And so what happens if then what we can do is we can take a look for each one of these clones or states, what is enriched, what are the cell types that are enriched in their neighbors. And so here are immune cells and macrophages and plasma cells. And blue here means depleted and red means enriched in their local neighborhood. And you can see, for example, that this particular for example, CD8 T cells are enriched next to one particular clone and transcriptional state of tumor cell and depleted from another. And we actually also then sequenced the T cell receptor sequences of these CD8 T cells. And it actually revealed that a particular clone of CD8 T cell is specifically interacting with this particular tumor subclone. And actually, this is one of the first times we were able to observe CD8 T cell clonality linked to specific tumor genetic clone. And we spent some time thinking about what was the mechanism driving this. And there's two possible reasons. One is that this particular tumor 1 state is losing its melanocytic identity and associated antigens that that CD8 clone could potentially recognize, or maybe it's losing its melanocytic identity and suppressing have some immunosuppressive program. Here we perform some epigenetic sequencing, and we actually show that basically there's a cell state transition towards a mesenchymal-like phenotype in this tumor 1 clone, and you can actually see that in the spatial data. If you just score, here's tumor 1, which is that which is depleted from that particular T cell state. It has adopted this mesenchymal-like phenotype, and it actually corresponds to, there are particular TF motifs associated with that cell state transition. And to make a long story short, some of these TF motifs are actually correlated, actually also clustered together spatially, like June and these IRFs, which suggests that maybe there's some local microenvironmental pressures or maybe epigenetic inheritance through cell division that's forming these little niches that allow perhaps, pushing the tumor to this dedifferentiated state, and also excludes that particular T cell clone from engaging in it. So this is an example of differences in cellular organization driven by disease context. And then I just wanted to highlight communications, right? Can this spatial organization reveal not just like changes in tissue state, but communications between cells? And here, I want to highlight this particular example in growth factor signaling in cancer. And recently, we published this interesting story on growth factor signaling in Hodgkin lymphoma, which is a very particular, interesting cancer type. In B-cell lymphomas, for normal B-cell growth and survival depends on kind of signals from their environment, whether they have the right cytokines or the right antigens. And in malignancies, these cells have to overcome this dependence. And one way they can do it is by constitutive mutations in these signaling pathways. Another way is through exploiting signals from the microenvironment. And in Hodgkin's is a fascinating example because the tumor cells are kind of these large multinucleated cells, and they only make up 1 to 2% of the tissue, and they're surrounded by this unique microenvironment of immune cells, including many CD4 T cells and macrophages. So they must also have to suppress them in some way. And actually, Vignesh, postdoc in Todd Golub's lab, had this hypothesis that there are microenvironmental derived factors that actually are being basically co-opted by the tumor for cell survival. And so to explore this, we actually collected spatial transcriptomic data and slide tags from about a dozen Hodgkin lymphoma samples. And we conducted this analysis leveraging cellular context to uncover paracrine interactions. And so how did we do that? So it's actually pretty simple. Let's say if a receptor, it's like this green dot, is expressed by a tumor cell and ligands are expressed by nearby cells, their spatial proximity could imply a potential interaction. Conversely, even if ligands and receptors are highly expressed, but they're not locally co-localized, it's less meaningful. And so then we can try to enrich for interactions which are active. And one cool thing is that this leverages spatial heterogeneity in the neighborhood context. So it's akin to almost like a natural experiment in the human sample where the experiment is like perturbing who your neighbors are. And when you do that, you can see spatial enrichment of ligand receptor interactions shown in this plot where on the over here is basically ligands which are not spatially co-localized. Over here are ligands which are spatially localized, and up here is the expression. And so you can see many factors are highly expressed in the tissue, but maybe they're not spatially co-localized. But of these spatially co-localized guys, we find many strong environmental ligand signaling to tumor cell receptors, including many immunosuppressive and growth factors. Actually, we went and validated many of them with functional experiments. I'll just highlight one, IL-13 and IL-13 receptor, which is important because IL-13 actually has a lot of therapeutic antibodies that block IL-13 interacting with the receptor, mainly for autoimmunity indications. And it turns out that when you use these same therapeutic antibodies, you can block tumor cell growth both in vitro and in vivo. And so at really clinically relevant concentration. So it's actually like it's very possible that blocking this environmental signal for tumor growth, it's like the environmental dependency. And so, it's interesting because, leveraging the intrinsic spatial heterogeneity within the human samples, you can identify these paracrine growth signals critical for tumor cell survival. And that led to some testable therapeutic hypotheses. And so, these are just a couple of examples, right? But actually, I think this idea that changes in spatial context, whether they're neighborhoods, interaction, or signaling, are natural perturbational experiments within tissues, even human samples, is a pretty powerful one. And as we collect a lot more spatial transcriptomic data, we are leveraging that to generate hypotheses about how cells communicate and are organized inside of tissues. And in general, the group is trying to do that by developing new technologies to capture heterogeneity in somatic and epigenome variation. This is something that we're funded by this NHGRI mechanism to do, and we've really been also interested in scaling the ability to perform these sorts of measurements at scale in large scale experiments, and then also manipulating these communities and tissue organization in vivo. So I'll just thank the folks who made the lab the work possible. Jackson Ware and Andrew Russo, who did the slide tags work, and then some folks who have been working on the spatial reconstruction and methylation data, Chinley and Benno, and then collaborators Evan McCosco, Todd, and Vignesh. Thank you.
Thank you, Dr. Chen, for that fascinating talk. How does your assay compare to the StereoSeq, which is nanoscale spatial transcriptomics?
I think the main thing is that for tags, you're just sequencing you're performing single cell sequencing, and the main caveat there is that the main advantage is that it's true single cell information. There's no segmentation. You're measuring the whole transcriptome. It's generally much higher quality data in terms of number of UMIs per cell, and the degree of mixing that you see. slide-seq is very much similar to StereoSeq, actually, because you have individual pixels and then you capture the mRNA. These days, we're excited about tags just in the sense that we think there are a lot of analyses that really need like true single cell resolution and unbiased measurements. Yeah. And then there's another question about receptor binding communications. Is the data for highly expressed, highly localized expression corrected for ligand receptor binding strength on off rates? No, it's not. And that's a good question. We do not correct for that. Mainly also because we don't have great measurements of that for lots and lots of receptors and ligands. And that would be something that would be interesting to collect and integrate into computational analyses. Another thing would be like if there were transcriptional responses to ligands that could be integrated.
And I have a question for both of you. What do you envision will be the next breakthrough in spatial profiling? We've already come a long way. And when looking at current technologies, what limitations do you foresee being overcome like in the next 5 years? We could hear both your perspectives on that.
I guess on the RNA front, I feel like technologies are pretty mature. I mean, there's lots of commercialized technologies that allow you to measure transcriptomes to varying degrees. I think there's a couple of challenges. One is, to what degree are those transcriptomes, like to what degree can you collect those without a paired single cell reference? Generally that has not necessarily spatial has not replaced single cell, although in theory you should be able to, right? And probably that's because one, you don't measure all the genes, and two, there's mixing in the data. So that but I think that can be reasonably addressed. So I think on RNA, I think it's quite mature on data collection. Then of course there's like having the right thinking about what are the right questions you want to ask the data. Like most questions people are thinking about revolve around cell types versus gene expression programs or changes in gene expression in those cell types. Um, and so just thinking about what are questions that you could actually answer with spatial context that's beyond just the organization of cell types is interesting. And then obviously there's molecules that are not RNA, which are much harder to measure, like the DNA and epigenome. I also think that will probably come in the next couple of years too.
Thank you. So maybe, I agree and disagree a little bit, right? I agree that RNA has definitely became mature technology in the sense it commercialized and sort of reached some sort of plateau. I'm not sure that plateau is necessarily satisfying, right? I think the cost sort of per sample and sort of the number of transcripts we're measuring per sample and so on are just too low, right? To actually really unlock the power of sort of spatial biology, right? And then maybe Fei was kind of correctly, right? We're still very much tethered to sort of single-cell RNA-seq, right? Although I'm not sure that that's tethered necessarily bad, right? The advantage of single-cell RNA-seq is that you can sample, right? You don't need to measure everything, right? So to determine the next sort of, predict the president of the United States, right? So it's 300 million people in the country. You only give a call for 1,000, right? You sample random 1,000 people and you get a pretty good prediction of what will happen, right? So we don't necessarily need to measure everything if we can sample properly. So that's the advantage of single-cell RNA-seq that I think sort of will always make sort of you want to still take advantage of, right, when you're doing spatial, right? The ability to sample, get a good information, and then sort of move that information into space.
Okay, go ahead. But you can
And then
sample
I
in
just want
space
to say
too.
that I completely agree with you. It's sort of trying to think more, right? We really need a whole new language of how do we talk about organization and communication and integration of sort of physical models, biophysics into all of this is really essential to sort of move the frontier in terms of sort of our modeling and thinking about all those tools.
Oh, yeah, I was just gonna say you could take your argument further though. You could just sample in space. If the data was truly the same, if the data was truly the
Yeah,
same
but it's not,
quality,
right?
then you would
But
just
that's the whole
sample in
issue, right?
space.
Like it's
Just
not true, right? You have it's stratified, right? I can't
yeah,
actually get everything, right?
yeah. So that was more my point. That
Yeah.
you could still get away with it if you thought that the
Yeah.
data was the same.
Yeah. But I mean, I think spatially, right? When you're randomly selecting, you're capturing all regions with whatever
Yeah.
probability. Whereas where you're measuring a section, it could easily be that the section just above it is completely different.
Mm-hmm.
Right? And you have no way to know, right? Whereas you're sampling randomly, you do capture sort of some representation of the entire distribution.
Mm-hmm. Yeah, that's true.